All right, well, hello and welcome to the Alaska Sea Life Center once again for another Telequarium. Hopefully you caught us a couple of hours ago when Maddie was leading stretches in front of the sea lions, but now we're going to have something a little bit different. I'm kind of in a strange room here. This is what we call our changing exhibits room. And as the name implies, the exhibits frequently change in here. But this year we decided let's make this room a focus of our science at the Sea Life Center. Because the Sea Life Center, we do have a public-facing aquarium, but a lot of our work is kind of behind the scenes, down in the labs, doing research. And people don't know about that a lot of times if all they're doing is seeing the front of house. So we've created some exhibitry in here just talking about our research, and I'm in front of this big wooden cutout of a shark. And I'm not sure if you can see up there, but it is a Pacific sleeper shark. And this is something that we've been doing some research on the past few years now. And today we're actually going to get a talk from Dr. Amy Bishop. And she is going to be kind of walking us through some of the research that she's been doing with the rest of our research team here at the Alaska Sea Life Center. So we're going to hand it on over to her, and we'll get started in just a moment.
All right. Hello, everyone. As Alex said, my name is Dr. Amy Bishop, and I am working from home, as many of you might be in these times. But I'm really excited today to be able to talk to you and share with you a little bit about this research project that I've been a part of for the last couple years. As you can see here on the screen, it is a big team effort, and it involves scientists at the Alaska Sea Life Center, as well as our partners at California State University, Long Beach, the Shark Lab. This project is funded by the North Pacific Research Board, and the goal of it is really to understand this species, the Pacific sleeper shark, that we don't know a whole lot about. So to jump right in, today I'm actually going to take a couple steps back, and this story actually begins a long time ago, and for many of you in an ocean far, far away. And the story begins with stellar sea lions. So stellar sea lions, for those of you who don't know, are a pinniped that are found here in the coast of Alaska, as well as along the Pacific coast of the United States and Canada. And the population is split into kind of two groups based on genetics and other all behavioral and kind of space use patterns. So as you can see in this map here, there's a line, and there's kind of the dashed region that's most in Alaska, and then the dotted region that's along Canada and the East Coast. The part that's mainly in Alaska is called the Western Distinct Population Segment. Another reason why we have this differentiation between those two populations is that we've seen a very different trajectory in their populations in the past 40 or 50 years. So right around 1970, 1980, we saw this really big decline in stellar sea lion populations in that western stock, the Alaskan stock of stellar sea lions. And during that time, a lot of people, including this great team of scientists from the Alaska Sea Life Center, started to try to develop research programs, ask questions to try to figure out why that decline happened, and perhaps why the population wasn't showing any signs of recovery in more current times. So in the early 2000s, this team got together, and in particular, Dr. Marcus Horning, who is one of our senior scientists here at the Alaska Sea Life Center, was really struck by the questions of wanting to know how long individual sea lions live, where they die, and how they die. And in particular, we were interested in juvenile stellar sea lions. So those are animals that have weaned, they're no longer relying on their mom, but they haven't quite hit adulthood. They're not part of the reproductive age group yet. And that's usually an age class that's pretty vulnerable for most species. That's where you see a lot of mortality events. But when you're working in a marine environment, it's really hard to be able to track what's happening to animals, especially animals that spend a significant part of their life underwater. So Dr. Horning actually, with colleagues, designed a new type of telemetry tag called a life history tag. It's these pill-shaped tags that you see in this picture here. And they're actually implanted into animals using standard surgery techniques. It's kind of like a pacemaker. And it sits in the animal's body cavity, and it records their temperature throughout their whole life. That's a great way for us to know, one, what's happening with the animal, is it giving birth when it hits adulthood, and that sort of thing. But it also lets us know about mortalities. When an animal dies, that tag will eventually emerge from the animal. And so whether that's because the animal died and it decomposed and the tag comes out, or if there was something more violent like a predation event where the tag might come out very rapidly. The tags float, so then they come to the surface of the water, or if they're on a beach because the animal died on land, we wait for it to decompose, it's exposed to the air, and it starts transmitting all of that data that it collected to satellites. So we can get data from animals many years after we've put these tags into them. So, 5, 10, 15 years, depending on the battery life, we can still get to know what happened to these animals. Now, that temperature data I talked about is really where all of this started. So doing some standardized testing, we were able to see that for tags that went from kind of happy warm sea lion temperature down to air or water really fast, that's the kind of profile we would expect. So that's something we might expect if an animal was predated upon and the tag was very rapidly extruded from the body before the body had a chance to cool. Similarly, modeling what would happen if that same kind of sized animal, a 70 kilogram animal or a 184 kilogram animal, died by more natural causes, so maybe disease or malnutrition, and then the body just normally cooled as it would. So this is that kind of temperature profile we would expect from a cooling type of death. Well, our initial findings, we put LHX tags into 45 different juvenile animals in the area. And within the last 20 years, we've had 18 mortality events. So that's almost, sorry, there's actually 20. This is an old slide. 18 predation events. And they've happened all across the Gulf of Alaska, as you can see here. And from that temperature data, we found that 18 of those mortality events were related to predation. And you can see here some of that data in real time. So this is, again, those air and water profiles that we kind of simulated or tested. And this is the data from the actual animals. So you can tell they went from happy, warm, sea lion, normal core mammalian body temperature to really cold air and water temperatures very rapidly. So this really let us know quite a bit about what was potentially causing mortality for this age class. Now, if we go back to that kind of temperature profile that we were looking at before, we noticed that for a couple of the animals, we saw that rapid cooling that we would expect from predation, but one of the other sensors on the tags is actually a light sensor. So it's able to tell if the tag has been exposed to light, which you would only expect to happen if the tag had been extruded from the body, because once it's in the body, it's dark in there. So in these cases, the tags went really cold, but the light sensors did not trigger, which, thinking about potential explanations for that pattern in the data, our most parsimonious or our best solution or explanation for that data is that some cold-blooded animal, most likely ate or predated upon the sea lion, ingested the tag, and then, as you can see in that plot, about 11 days later, 5 to 9 days later, that light sensor triggered, which was likely because the shark either regurgitated or defecated the tag. When we look at the shark species that are present in Alaska, the only one that makes sense for this really is the Pacific sleeper shark. So that's the nutshell of why we, mainly a group of stellar sea lion and pinniped researchers, started to get really excited and interested in this idea of sleeper sharks, because we wanted to know more about all of these interactions between predators and prey in the Gulf of Alaska. This is a picture of a Pacific sleeper shark, one of the little ones that we worked with as part of this study. Their Latin name is Somniosus pacificus. Somniosus means to dream or sleepy, which makes sense because these animals are, as the title of this presentation said, very, very slow moving and long-lived. And that's really one of the only things we know about Pacific sleeper sharks. We know that in Alaska, they are caught as bycatch, so incidental catch, in a lot of Alaskan fisheries. This is some data from a report that was done in the mid-2000s that shows different shark species that are accidentally caught by fishing operations in Alaska. And Pacific sleeper sharks are the gray squares that you see here. So they're caught quite oftenly by commercial fishing operations. So we know that not only do they have an economic impact on the fisheries by being incidentally caught, but they're also being removed from the ecosystem. So understanding more about their biology can both help us understand how to manage fisheries and be able to help them, but also understand what role they play in the ecosystem. And that's what's really being done a lot more in these ocean ecosystems, is we're really interested in understanding how all these pieces fit together. So how the actual fishing operations linked to the communities that rely on them and linked to the animals and the ecosystems that they're living off of and utilizing, and how all of that fits in together as part of our fisheries management. So this information about this species can be really important for those sustainable, healthy ocean ecosystems. Otherwise, we don't know a whole ton about the specific sleeper sharks. There haven't been that many studies on them. More recently, though, there have been a lot of studies, and you may have heard in the news about Greenland sharks. They are a closely related species to the Pacific sleeper shark, and they hit the news a couple years ago because a study found that by aging these animals using their eye lenses, they estimated that some of the largest animals that they sampled actually can live 250 to 500 years old. Let that sink in a little bit. There are potentially 300-400-year-old sharks swimming around in the ocean. And because Pacific sleeper sharks are so closely related to Greenland sharks that in the Arctic, they actually hybridize, we have a good inkling or a good set of evidence to suggest that it's likely Pacific sleeper sharks also have a long lifespan similar to the Greenland sharks. So they're interesting for fisheries management and ecosystem management. They're interesting for the role that they play with stellar sea lions in this kind of predator-prey dynamic for an endangered species. And they're just really cool because they live to be so long. So our researchers were super excited to start to explore the species and try to understand just some of that basic information about it that could help us develop new questions that we could test that could relate to those kind of three facets that I just so this map here is of Alaska. For those of you who aren't familiar, the red dot is roughly where Seward is on the Gulf of Alaska coast. This is a zoom in, so the Glasgow Sea Life Center is now the red dot, and this is a picture of Resurrection Bay. It's the fjord that Seward sits at the mouth, or sorry, the head of, and most of our effort was actually done right here. We had talked to a local fisherman who told us, nope, you can totally catch sleeper sharks right here in the bay. So we took him at his word. We were funded by, like I said, the North Pacific Research Board. And in 2018, we set out to try to find some sleeper sharks. This is our research boat, the Jubatus. And we set out in May of 2018 to start our research. That summer, it was quite wet and quite cold. So the days on the water are always great for us researchers. It's great to get out of the office, but it's not always pleasant as far as the environment that we have to work in. But we were all excited because, like I said, this was new territory for all of us. Our first couple days, we tried fishing with rod and reel, and finding that to be less than successful. We kind of reevaluated our methods, and we moved to this approach where we had modified long lines. So basically, we had a buoy with a set of line that at the bottom of it had two hooks suspended by gangins and an anchor. And we used bait from local fish processors like salmon or any halibut that we accidentally caught. So this system, basically we would go and set these buoys, 4 of them kind of spaced out across a set area in the bay. We'd let them soak for about 6 to 7 hours, and then we'd come back in the afternoon and see what we caught. And for the first couple months, we didn't catch a whole heck of a lot. We caught, as you can see here, accidental, again, bycatch. We caught some species that we weren't targeting, like halibut. Though this halibut, we had a sinking suspicion that maybe a sleeper shark had come by and taken a bite out of while it was on the hook. But those first couple days, we did spend a lot of time kind of waiting for sharks. And so as part of that, we started thinking, well, maybe there's some other things that we could do to increase our likelihood of catching sleeper sharks. So we turned to the experts.
You see, Bert, I have to call them really loud, though, so they can hear.
Yeah.
The fish are under the water.
Sure.
And so you have to call real loud so they can hear you through the water. You know?
Sure.
Okay.
Go ahead.
I call them like this. Fishy, fishy, fishy! One.
So knowing that worked so well, we thought we'd give it a try.
Fishy, 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 fishy!
Needless to say, we basically did that rallying call at the beginning of every single one of our sets until finally in early June in 2018, we started hauling in our line, and you can see here what that operation looks like. And as we pulled that line in, we started to realize it was really heavy. We were blowing fuses left and right. We had never really had that happen before. And sure enough, you'll have to tune in next week to see what came to the surface. I know, a little mean, but this is such a great story, and there's so much additional data that I want to share with you and talk to you about that I just couldn't do it all in 20 minutes. And I really wanted to give you guys the chance to learn about why we were interested in sleeper sharks first, learn a little bit about all of what we did with the data from the LHX tags, and hope that you all tune in next week to find out what we've learned about this amazing species. And if you have any questions, I'm just going to quick throw up again our permits and funders for both the sea lion research and the Pacific sleeper shark research. And I think Alex has been monitoring if anyone has any questions online. I'm happy to answer them now, or like I said, tune in next week and gather your questions and I'd be happy to answer them then.
I think I was muted there. I was muted. No one's got any questions for you in the chat right now, but even for the folks that may be watching this not live, that's okay. Go ahead and type in any comments that you want down below with questions for Amy or anyone in the research project. Honestly, we could reach out and try to get those answered for you. But like Amy said, tune in next week because it's pretty exciting once they actually have the shark. You know, like this whole science of how did we even think maybe the sharks were involved? That's fascinating to me, just looking at numbers, right? And some of those numbers, for whatever reason, they seem a little sharky, is kind of what happens here, right? So science is constantly full of unexpected things. You're looking at sea lions, you're watching their life, and then all of a sudden you go, wait a minute, how did that sea lion die? And when you start thinking about that, it brings up very strange things, like perhaps these sleeper sharks. Amy, my favorite thing about them probably has to be that lifespan. And obviously you said that was with Greenland sharks. Have we been able to, or is there any research to kind of do comparable studies of age with the sleeper sharks versus the Greenland sharks? Have we been able to confirm any old sharks?
Yeah, so that is a great question. Some of our collaborators at the Alaska Fisheries Science Center at NOAA, they are trying to look at things like age and growth for these animals. They're actually using those sharks that are accidentally caught in commercial fishing operations, and they can biosample them because a lot of times these animals don't survive when they're caught and they're kept on the hooks for a long time or just in the handling process. And we'll talk a little bit more about that next week, about some of the measures that we put in place to try to make sure that we maximized, or in this case, minimized any damage to the animals. But when that does happen, it gives us a great opportunity to still learn about these sharks. So researchers, like I said, at NOAA are collecting biosamples from those animals. And for sharks, because they're cartilaginous, meaning that they don't have bones like we as humans do, they have cartilage skeletons. We can't use their bones or their teeth to age them as we would with maybe a mammal. So what they have to use is the eye lens, and they can use it by radiocarbon dating it. And so those researchers are collecting samples to start to look at some of the ages for Pacific sleeper sharks in Alaska. And one of the things that we do, giving away a little bit of a preview of next week, one of the things that we do when we do catch a shark is take measurements of their length, their girth, and different measurements throughout their body that hopefully will give us ways that once we get some of that aging data, we can relate the growth rates and the morphometrics, their sizes, to age, so that we would get a better idea of how old an animal is just by measuring it. Right now, based on the Greenland shark data that we have, most of the animals that we handled were anywhere from, I should use metric because it's scientific, but anywhere from 5 to 12 feet. And we're estimating that those animals were probably in the range of, 40 to 80 years old. And they are technically considered juveniles at that age. They're immature.
Very cool. And obviously everyone should tune in next week for another Telequarium where we talk to you. But you do actually document some of the science going on here at the Sea Life Center on a blog as well.
Yes.
If you want to point people that way.
Yeah. So we have a science blog that I do a lot of the editing and writing for, but we also have great other contributors, grad students from around the country talk about their research there. We try to update it once a week with some new content. So definitely click on that, subscribe if you want to get some more information. And a lot of our research that we have done on sleeper sharks has been already documented there. And there's some great stories, videos, and pictures if you're interested.
Perfect. I actually do see one question that popped up in the chat here really quick. Someone asks, do you all take graduate student interns for our research team here?
We don't take graduate students at the Alaska Sea Life Center because most of our researchers are not affiliated with the university directly, but our collaborators do. So on this project, Taylor Smith is a master's student from Cal State University in Long Beach, and so she is doing a lot of the metabolic and energetic research as part of her graduate studies. We do at the Sea Life Center, though, take interns. And so those are great opportunities for early career students and researchers to get hands-on experience in the field, to be part of these research teams, to learn about data collection, data analysis. We usually take 1 to 2 each summer, and those opportunities can be found on the Alaska Sea Life Center webpage. If you are specifically interested in other graduate student opportunities, I still encourage you to reach out to us because, like I said, we may not be able to directly take you on as a grad student, but often our projects and our collaborators have opportunities for graduate students to participate.
Yeah, well, thank you so much for talking to us about this. I like this little bit of a tease about next week because it's like fishing for sharks. It's cool to be like, oh yeah, we're trying to catch these sharks for research. It's really cool when the shark is actually there. So I hope everyone will tune in next week. And thank you so much, Amy, for telling us about this work. Yeah. And we'll see everyone, hopefully for our future Telequarium. We're doing live streams 12 and 2 every day on our YouTube. We're also getting some over on our Facebook, but every day, 12 and 2 on our YouTube channel. We're doing these Telequariums where we're bringing the Alaska Sea Life Center to you over the vastness of the internet, and hopefully we'll see you for more of them. So thank you so much for tuning in.